Hello, my name is Homayoun Valafar, and I'm a professor of computer science and engineering at the University of South Carolina. I direct the bioinformatics core of the IMBRI project throughout the state of South Carolina, and I am the director of the genomic core of the Big Data Health Sciences Center at USC. Today, I would like to share with you some of our latest work related to the study of COVID-19. Since its appearance in 2019, SARS-CoV-2 has been identified as the cause of the COVID-19 disease that causes respiratory and cardiovascular complications. Thanks to modern advances in biotechnology and imaging technology, scientists around the world have a relatively complete understanding of the topology of this particle and the mechanism of its entry to the host cell. In particular, the spike protein of SARS-CoV-2 has been identified in its binding to the ACE2 proteins, which are abundant on the surface of the lung tissue cells. Furthermore, thanks to the advances in genomic sequencing technology, combined with bioinformatics techniques, we now have completed the genomic sequence of the SARS-CoV-2. While we have a relatively complete understanding of the mechanism of the infection outside of the cell, very little is known about the viral life cycle inside the host cell. To better understand the virus's life cycle, we have targeted the study of the protein called non-structural protein 1. Non-structural protein 1, or NSP1, is an intrinsically disordered protein that has been implicated as one of the first steps in establishing the life cycle of this virus. NSP1 executes a two-pronged approach in overtaking the host cell's functions. First, it disables the synthesis of the host cell's proteins, therefore shutting off the host's immune response. Second, it prioritizes the production of the viral proteins by hijacking the protein synthesis machinery of the host. Using computational modeling techniques combined with deep mining of the protein data bank, we have developed a functional model of this protein. We have also developed our own computational analysis pipeline that integrates evolutionary and data mining searches to identify the functionally important regions of this protein. Using our analysis pipeline, we have identified the region that's shown in green, which establishes the first contact with the ribosomal complex within the host cell. We have also identified a second critical binding site of NSP1 that is located on the unstructured region of this protein. This region is also illustrated in green among the unstructured region that's also depicted in red. This second recognition site will help to mold the flexible portion of the protein around NSP1's target to establish a firm grip and to hijack the protein synthesis function of the host cell. We are currently using this functional basis to develop mechanisms of disrupting the life cycle of SARS-CoV-2 in our combat against this disease. I would like to thank my collaborators, Dr. Anita Nag at USC Upstate and Dr. Casey Cole at USC. I would like to acknowledge my students' help, Niharika Pandela and Devon McFarland, in performing the molecular dynamics simulations, folding of the protein, and other studies. I'd like to acknowledge the funding source of Office of Vice President of Research at USC. I would also like to acknowledge BioLotion for the video that they have posted on YouTube. Thank you.